So thanks for registering for the 6 days online FDP on tailored treatment in advancing faculty knowledge in personalized medicine. So I personally thank the ACD ATAL for giving me the opportunity to conduct this 6 days seminar, 6 days faculty development program. Next, I thank the management, staffs, and my co-organizer, Dr. Preeti Rebecca from Department of Computer Science, to coordinate and organize these events. So this will be a 6 days faculty development program. Today we'll be having 2 sessions. Tomorrow's 2 sessions. On Wednesday there will be a 3-session program. So followed by Saturday we have a 3-session on different areas of personalized medicine. Okay. To start with, today's session will be handled by Dr. Varsha Sridhar, who is a director co-founder of Molecular Solution Healthcare. So followed by Preeti Rebecca. So for each session there will be a feedback form. Please fill the feedback form. That's the main thing. So for every session there will be feedback form. Come and fill the feedback form. So tomorrow's session will be by Dr. Gopal Shankar from NID Calicut, followed by Ashwin Sai Narayan from NCBS. So Gopal Shankar will be talking about the 3D bioprinting, and Ashwin Sai Narayan will be talking about microbial genomics. So these two will be pretty interesting. So on Wednesday, there will be a session on morning. So it starts at 10:30 to 12 p.m. So it starts at 10 a.m. Please join for this session. This will be on oncology. So this speaker is from US, so we couldn't able to accommodate his timing. So it's better to join in the morning. So we organize the timing on. So please make sure that you join for this session. It will be really, really interesting. For my session though, I'll be giving a talk on toxins on different channels. So then we have Kirtivasan. So he'll be giving a talk on drug discovery. So he's a director from Alvotech. So he'll be giving a talk on drug discovery programs. So how to manage the drug discovery programs. So the next session, this is on Thursday. The Thursday session will be given by Pavitra Venkatagopalan. So this will be very interesting, where we'll be working on the new different strains. How she has worked on the new different. I think you should have seen her in many TVs during the COVID time. So she's the one who is going to give on different strains of COVID and what are all the new strains that comes, different viral strain. Followed by Shevli Ghosh. So she'll be giving on how the bioinformatics will play an important role in personalized medicine. So on Friday session it will be handled by Thomas, who is from Denmark. So he'll be giving on biofilms. So how the biofilms will play an important role in creating new drugs and personalized medicine. Followed by Dr. Rajesh, who is from Anna University Trichirappalli campus. So he'll be giving on production of antibiotics and pharmaceuticals, where it could play a very important role in pharmaceutical industries. So lastly, on Saturday session, this is also 3 sessions where one will be handled by Dr. Suryapriya Karnagaran. So that will be a morning session. So that will be a morning session where she'll be giving how to commercialize the biological drug product. How that will be commercialized. So followed by Sudarshana Deepa. So she's from NIT Andhra. So she'll be giving a view on how the nanobiotechnology will play a very important role in drug discovery and then various fields of medicines. Lastly, it's the last session is 5:15. That's on Saturday. So this will be handled by Dr. Indumati Sasisaran. So she'll be giving on how the drug is formulated for a personalized medicine or genomic medicine. I hope this audience is a mix of computer science and biotechnology background. So please try to understand and please make it more interactive, ask more questions, so they might be helpful for you in your collaboration, or they'll be helpful for your PhD thesis or PhD work. So they'll be helpful in your, so most of them are my friends, colleagues, my students. So these are like, so this will be a platform where you can have collaboration.
The like personalized medicine only because every individual has their own genome sets and every person in this earth is different from each other. So there the medicine will not fit everyone. So we need to identify this kind of methods to prevent the diseases. So genetic profiling is one of the most important factor in personalized medicine. Next.
There is a specialized area called biopharmaceutical industry, which is developing biologics. And these are not as simple as developing a tablet. These are complex products, and these are produced by living organisms. So to give some example, let's start with this. So I like this figure. So a small molecule drug takes comparatively less time to develop. It's produced by a chemical synthesis route. It's a very small molecule, like aspirin, for example, has 21 atoms. So this can be like compared to a small boat.
Grow them on a flat surface, and the cells are flat and do not represent your real cell physiological morphology. All the cells in your body are growing in three-dimensional. But when you do it in 2D culture, they are not in three-dimensional. They are interacting with only one particular phase. Other two phases are left exposed. So 3D culture is the best place, that is a 3D space that is more essential for cell growth. And when you do that, the cells will form aggregates, or you call them as preoids, and they have more space for their interactions. So here you see the cell-to-cell contact interaction happens, the cell-to-scaffold interaction happens, and the cell-to-media interaction happens.
What happens is the property of hydroquinone and benzoquinone is combined to form a new crystal structure. This is called as co-crystal. Co-crystal is nothing but you will be having two different crystals which would be combined to form a completely new molecular entity. From this co-crystal only, crystal engineering approach got evolved. Now, basically, the slides, whatever I will be presenting, will focus on how we can improve the solubility of BCS class 2 and class 4 drugs, because these drugs possess poor solubility. And also BCS class 2 example, I have used carbamazepine. Finally, we will be studying the bioavailability studies using 3D cell culture models of triple negative breast cancer cells.
The number of bacteria on the planet is estimated to be somewhere in the 10 power 29, 10 power 30. The number of insects is believed to be about 10 power 18. Humans, close to 10 to the 10. So this is not just a majority, it's just that pretty much anything other than bacteria are a dot on this large space of cellular beings. They are the oldest forms of cellular life, most possibly, and by far the most dominant forms of life, and capable of an extraordinary range of metabolic and adaptive capabilities that other beings cannot do.
And this is basically how the early warning system works. Interestingly enough, a lot of the viruses that are actually respiratory also infect the gastrointestinal tract. So this you see, of course, in COVID, because you remember in the early days of COVID, people would say you lose your sense of taste and smell, but you also might get diarrhea. So basically, it is the virus infecting cells of the gastrointestinal system, which is the reason why you get diarrhea, right? And it goes through the wastewater system into your open drains or your sewage treatment plants and all that stuff. But it's not just SARS-CoV-2. Influenza does this, adenovirus does this. A lot of infections, basically, especially viruses, not so much bacteria, because they are a little more restricted in their host receptors, but many viruses have the potential to infect multiple types of cells, just because their receptors are a little more broader.
Medicines will be looking into the big data analytics, sequencing, etc., all these computer science part of it. Whereas this personalized medicine Initially, we'll be interviewing people on what actually is the symptom they have, their backgrounds and all other informations, one-on-one on a patient or a group of people, and then then go in for the next part of their drug discovery. And the end goal of both these things, that is, the end goal of traditional medicine will be you give them an optimized treatment that usually goes for a group of people who will be sharing same kind of a biological or genetical similarities. Whereas when you say it's a personalized medicine, you are more specific. You're more specific to individuals. So this way, the traditional medicine systems that we are presently following varies from the personalized medicine system, which we are actually trying to move forward to.
So what is the speciality of venom? So venom is a complex molecule. So venom is a complex molecule. It has lots of small molecules. It has lots of proteins. It has lots of peptides. So these peptides can be the target for particular sodium channel. So likewise, we started to work with Rim and Balkis from Institute of Pasteur, Tunis. So this was an Indo-Tunisian bilateral project where we took thing from scorpions, the venom from scorpions, because that LVMT lab in Tunis is the lab which produces anti-venom for all the scorpion toxins all over the world. That's the only lab, approximately 80 to 90% of the anti-venom that is being produced or that is being used are from this University of Pasteur lab.
When you design a sequence, or when you're working with proteins, let's say you're working with a CRISPR protein, or you're working with, you take a sequence from bacteria, you study about it, like, or many of the CRISPR molecules is from bacteria. But how do you make that work in humans in a way that it doesn't trigger silencing or trigger rejection? How do you make sure that foreign sequences actually work in the host organism? And that's the way usually you approach that is codon optimization. And I applied the same approach of using generative models to coding sequence optimization as well. How do you change the codons in a way that a foreign sequence now is understood as a host sequence?
So instead of looking just one aspect of the disease, we combine gene expression data, protein-protein interaction, metabolic pathways. Okay. So in gene expression data, what do? We identify which genes are up-regulated and down-regulated. And in protein-protein interaction, which proteins work together for that particular disease. And metabolic pathway is about how does the disease affect the body functions. Okay, so these are all the important aspects where you will be where you will be designing how this personalized medicine is important to the designing for this holistic disease, for designing a holistic disease modeling. Okay, this and next is the
the accepting me to attend this wonderful, incredible, this FDP program. First of all, I would like to express my gratitude to the organizer for selecting such important topic, tailored treatment, advancing faculty knowledge in personalized medicine. For the 6 days, which is sponsored by AICT, this ATAL program. So, extend my sincere thanks to Professor Kauri Sanga, Professor Andhat, who organized this wonderful event. I have almost attended all events, but I can tell you some topic which is attract to me, especially the yesterday who Tom 7 years actually from RS University, who have the presenting talk about the role of eRNA in biofilm. So even though the biofilm is very important, but he described how this eRNA which involved for the regulation of biofilm, future this eRNA may use for therapeutic purpose. Then another important lecture which delivered by Dr. Pavitra, who discussed about the viruses, how this impact in human life. Her insight definitely which is very informative and very useful. Additionally, Vardha Sridhar, actually she presented a wonderful talk about antimicrobial resistance, which is very important, every day, which is impact in our life. Then another lecture by Dr. Ashwin Sai Narayanan from NCPS, who talk about the evolution of microorganisms, how these microorganisms relate to one another in different environment, and discussed mostly the bacterial genomics by using next generation sequencing, how this is useful for in disease perspective. As well as another, there are several speakers, mostly from India as well as overseas, especially talk about the role of artificial intelligence in biotechnological research. I'm also kind of new to this field, but hopefully I believe it is highly beneficial to my career. Finally, I have to thank this organizer. Wonderful. All 6 days I'm also working in private engineering college. How this organization is not at all easy. Then I have to thank all these speakers and the organizer as well. Thank you, one and all. Thank you very much.
Thanks, Dr. Saravanan. It's been pressure that we can collaborate and we can have that's what.
Yes.
These are all some of the positive things that I take, because it's been 20 years that I spoke to all these speakers. So most of them I personally know them. So they said, once I said, they said I will give a talk. So once they gave the talk, it was interesting for the audience. Now I am planning for a big conclave where like-minded people can come, sit all over from the world, or like we can have a virtual or physical, where we can organize similar conferences, where it will be a good opportunity for them to give a feedback for us and us to give the feedback for them. So it's a mutual thing for collaboration, for research, for international conferences, for anything, whatever you call. That will be the starting point. So this will be the starting point for it.
Yes, definitely useful for us. Very useful. Hopefully, make a offline the regular meeting, the conference, whichever symposium. It create help help us to understand about especially the artificial intelligence. So in future, everything we are talking about artificial intelligence. So without artificial intelligence, I don't know how this impact to how very difficult to teach in biotechnology. So hopefully this FTP program definitely help. So I have a lot of ideas to do. So thanks, Gauri, for organizing wonderful, incredible event. Thank you. Yes.
Any other feedback? So I got lots of feedback in the chat. Thanks, Yashoda ma'am. Thanks, Dr. Sumati. Thanks, Pudhasman sir. I think, yeah. So there was a question from Parimala. Yeah. You can go ahead.
This is Roslyn, assistant professor. Hello, sir. Can you hear me?
Audible. It's audible.
Sir, this is Roslyn. Roslyn, assistant professor. So I attended this ATAL program, sir. It was really interesting, very informative. Various fields were covered in this FTP. So with no expectation, I just joined this event. I felt it was very informative. Most of the sessions were very new to me. I hope I will work further in my future on all these fields. Like, either at least any one of this field would really help me in my future research. Thank you for organizing such a wonderful event, sir. Preeti ma'am, thank you for the same. So hoping for further more events offline on such events could be organized so that you can have more gatherings, which can lead to, as you said, it can lead to good stages. Thank you, sir.